Hello everyone, my name is Noeen and I welcome all of you in our first session of round 2 for SCWIST Science Symposium 2021. Our first 3 sessions are about biotechnology and I will introduce what we are going to talk about just in a minute. So our speaker today is Gagan Gill. She is a PhD student from Wilfrid Laurier University, and we have 2 judges today with us. They both are accomplished scientists in their field. Dr. Anjana, who is the director and strategic business consultant, and also a consultant of ANRS Consulting Organization Canada, and Dr. Brittany. She is tetanus toxoid vaccine manufacturing expert from Sanofi Pasteur, Canada. And I would like to give land acknowledgement. We would like to take a moment and reflect on our connection to the land and thank the traditional guardians of that land, which we at SCWIST Vancouver live and work. Vancouver is on the unceded ancestral and traditional lands of Musqueam, Tsleil-Waututh, Seychelles, Squamish, and the indigenous people of Turtle Island. So as I already mentioned, today is the first session of our round 2 competition where all participants will give their presentations and then there will be panel discussions and question and answer. That's how they will be, their research projects will be evaluated. So today the first speaker is Gagan from Wilfrid Laurier University, and there are some housekeeping rules. Please note that today's event is going to be video recorded and you will keep your mobiles, your microphone muted all the time. If you have any question at any time, please type in the chat box. As I already mentioned that after presentation by speaker, which will be around 12 to 15 minutes, there will be panel discussion together with judges. And after that, there will be question and answers from the audience. Please type your question and answers in the chat box. We will ask during that time, and at the end we will have our closing remarks for today's session. This YouTube video will be uploaded to YouTube SCWIST channel by tonight, and I will share the link with our all finalists as well as our judges. Here are our 2 wonderful judges, and before I leave the floor to them for their introduction, I will just say about the title of Gagan, which is testing and application of RNA-Cleaving DNAzymes for bioavailable metal sensing. And this session today is proudly sponsored by Admire Bioinnovations, Microsoft, Equitas Therapeutics, Northeastern University, AbCellera, and Sixth Sense Strategy Group. Our sponsors are all they all offer lots of opportunities for our students. For example, Northeastern University, the employment rate for biotechnology is almost 95% for the graduates. You can check on their website. For Admire Bioinnovation, they offer programs in the form of Bioinnovations Academy. And if you want to polish your skills as a scientist or business, you should definitely like to explore that program list. And AbCellera, there is one big news that newly discovered antibody that neutralizes viral variants of COVID-19 is the new innovation from AbCellera. From Equitas Therapeutics, many of you don't know, but it's a very new and fastly growing company. You can see its importance from this fact that they provided their lipid nanoparticle delivery system, which is the key element in the development of Pfizer's vaccine for COVID-19. We have Microsoft here. Microsoft also provides lots of internship programs in information and technology, and Sixth Sense Strategy Group, which is basically a consultancy group providing strategies related to business in life science. So with this here, actually, I will stop sharing my screen and I will give the floor to Brittany first to introduce herself, and then to Anjana.
All right, good afternoon, everyone. So my name is Brittany Pequegnat. I have a PhD in chemistry from the University of Guelph, Ontario. I did a postdoc at Harvard Medical School in Boston. I then worked at a startup vaccine company, and I returned to Toronto, where I'm currently the tetanus toxoid manufacturing expert for Sanofi Pasteur, the vaccine company based out of Toronto, Ontario.
Thanks, Brittany. Anjana?
Hello, everyone. Good afternoon. My full name is Anjana Govindarajan. I have done a PhD in biotechnology. I did my PhD in India. Post that I moved to Nova Scotia. I worked at Dalhousie University as a postdoctoral fellow. Later, I got NSAC fellowship and worked at Agri-Food Agri-Canada. Then later on, with all these experience, now I'm an independent consultant. I do strategic business consultancy. Yeah. Welcome you all to the session, and I'm very much looking forward to have this discussion. Yeah.
Thanks, Anjana. So now the floor is to our speaker today, Gagan. Gagan, first you can give your introduction and then you can share your slides and start presentation. So it's all for you now. Thank you.
Sure.
Thanks, everyone.
Well, thank you very much to everybody, the judges, the audience, especially Noeen for organizing all of this and constantly being in touch. It's very easy to get a hold of her whenever you needed her. So thank you very much for all of that. As I've been introduced already, my name is Gagan. I am currently doing a PhD at Wilfrid University. I did a master's at University of Waterloo in nanoscience, chemistry as well, but specialized in nanoscience. I have a thing for sensors. My project back in my undergrad thesis was sensors. My project in my master's was also sensors. And then here I am back doing sensors again in my PhD. So I don't know what it is. I enjoy it. I think I've built some sort of love for it and what they can do and what they cannot do. So hopefully I give a good talk with you guys today. And then I'm looking forward to the panel discussion as well. Obviously, I love new ideas. So if you guys do have some of those, I'd be more than happy to take them. So with further ado, I'm going to start my presentation. I think I'm going to, I am double monitored here. So I just want to make sure that I am sharing the right screen and you're not seeing my notes. Share screen. Desktop too. Okay, so we can confirm that you guys are seeing this screen with no notes. All righty. Okay, so my presentation is application of RNA-Cleaving DNAzymes for bioavailable metal sensing. I know the title is testing, so testing application, same thing. My professors that I work with right now, Dr. Scott Smith, Juan Lu's from Waterloo. I actually work with my master's with him as well, so it's nice to kind of get back in touch. And then Dr. Scott Smith, I'm pretty sure he's in the audience watching me, judging me as well. So just gonna get right into it. So here's a table of contents. I'm gonna start off by talking about the novelty. I'm gonna get into my research purpose, the methodology. I'm gonna spend a little bit of time on there, explain exactly what I'm doing, how I'm doing it, so you guys kind of get a clear picture. Some results, not a lot of results that I'm going to get into, but just some results to kind of see what I'm getting out of my results, out of my experiments, and then the future picture of where I see my project actually going. So first things first, Canada is ranked number 8 in lead mining in the world. And I'm going to focus on lead for my project here specifically, because that's the starter that I've been doing, but it is generally a metals project. So New Brunswick, shown on the left-hand side over here, is the largest lead mining facility in all of Canada, and it mines thousands of tons of lead every single year. Actually, in 2020, because of COVID, the pollution of lead has decreased at a noticeable rate, thanks COVID, which halted the mining of lead for, I guess, 2020 and even part of 2021. The statistics that I got for lead mining was only from 2019 because they said that we don't have anything for 2020 right now. One of the main uses of lead is in lead acid batteries, but lead is also used various different, I think in paint is one of the other ones that it's also used in as well. Now, when lead enters into the environment through these many pathways shown in the diagram on the right-hand side, they can be found in different forms. They can be found in free lead, they can be found in lead-bound species, but depending on the species, lead that is bioavailable is presumed to be most toxic to its surrounding environment. It can be toxic to both people, and it's also toxic to aquatic species. Even in minor quantities. I think the toxicity of something like daphnia is less than 0.4 milligrams per liter for EC50s. Now, lakes around these mining facilities are filled with dangerous levels of lead. As you can see in the picture below, you can see the different lakes from acid mine from heavy metal contamination, from sediment and erosion, process chemical pollution. You can see all the lakes are all messed up because of this lead that's entering into the environment. But now, how do we monitor these levels of lead, and how can we control this problem? Now, currently, risk assessment managers use the biotic ligand model as a tool to assess and make decisions regarding these contaminated lakes. However, in order to do this, they must measure the inputs shown on the left-hand side using various instruments, and then they have to input these parameters into the model, apply it, and then predict bioavailability. I'll mention what bioavailability is in just a second. Now, these biotic ligand model inputs include temperature, so you have to measure the temperature independently, the pH, dissolved organic carbon. They have to measure the major cations, major anions, alkalinity, and sulfide. So these 7 major inputs that they have to measure independently at each site, implemented into the biotic ligand model, and then predict it. So it's a long process, and then at the end of the day, it's just a prediction. It's not even a measurement. Now, bioavailable metal is the portion of metal that's presumed to be most toxic. That's why I keep talking about bioavailable. I'm going to say it throughout my whole presentation. It's a tongue twister for me, so I apologize if I mispronounce it, but bioavailable metal. It's the presumed to be most toxic. What it is, essentially, it's a weakly complexed. So as you can see, the weakly complexed up here, plus the free metal ion over here, that is bioavailable. And essentially, this concentration of metal is what has the ability to attach to the biotic ligand of any species, and then have the potential to enter into the species and then be toxic towards them. So because of this, it's the bioavailable portion that is actually what is important for regulation purposes. The strong complex metal, which is shown by below, which is usually by chelation of dissolved organic matter or measured in dissolved organic carbon, so I interchanged those words there. That is not bioavailable, so it's not toxic. Now, speciation, which is weakly complexed or free, is dependent on modifying factors found in aquatic systems. So the most important ones are pH, ionic strength, and DOC, and those are the ones I'm going to focus on in my research. Others are major cations and anions, and all these individual parameters are currently have to be measured separately, like I mentioned. There is instrumentation that exists that can measure either the weakly complexed or the free metal or total metal, but nothing that measures the bioavailable directly. Now, after measurements, they implement this into the biotic ligand model, and then we predict it. And we focus on this metal, like I mentioned, throughout the rest of my experiments. So now here are the instrumentations that I mentioned that do exist, but they only measure the free metal, which is also the free metal, or the labile metal, which is also another word for a weakly complexed metal. However, the problem with all these is what we always go through. Usually, they're expensive, they're complicated, they're time-consuming, and they're so inconvenient. And there's no accepted method where you can measure the bioavailable metal directly. Now, instead of predicting the bioavailable directly through the biotic ligand model, we propose to measure that toxic fraction of metal directly, and we do this through biosensing. Now, we can do this. It's cheap. It's not complicated. It's not time-consuming. I get measurements within seconds. It's not inconvenient either, because I could do it at the point of care. I can go to the facilities I want to go to, and go to lakes that I want to go to, and be able to measure that there. That's the bigger picture. That's not where I am right now, but that's the bigger picture. So, like I said, what about biosensors? Now, what are biosensors? Biosensors is a self-contained integrated device, and it's capable of providing specific quantitative analytical information. You have a biorecognition element, which you can see here. I got DNAzymes, aptamers, and there's many other ones, and a transducing element, calorimetric, there's electrochemical, I worked electrochemical before, fluorescence. It's not necessarily a device, but it can be built into a device, a DNAzyme. Now, there's no existing biosensor that measures bioavailable mode directly. But by using DNAzymes as a potential applicant, we will apply the technology in a way that's never been applied before. So I want to emphasize DNAzymes already exist. Lots of them exist. I'm specifically taking the one that I want to use, which is specific for my metal, since my target is metal. And then in this case, my target is lead. So I chose this DNAzyme. It's called GR5. I'm sorry, it's not even on there. It's on the next slide. And then I use this technology in the way that I want to use it. Now, through this application, we propose that the DNAzymes will be excellent candidates for sensing this toxic portion of metal at the point of care. Now, what are DNAzymes? Shown on the picture on the left, exactly looks like a DNAzyme strand, double-stranded DNA. Basically, is what it looks like. It's not what it is, but it's what it looks like. They're single-stranded DNA-based catalysts that are obtained through in vitro selection, and for catalytic activities, they require metal cofactors to function. So the unique thing about these is that they actually need the metal in order to interact with that cleavage site, which I'll get into in a second, in order for it to experience catalytic activity. So in a way, the DNAzymes by themselves, without having to do anything with them, they're sensors on their own. Now, they have shown to have great sensitivity, selectivity, and speed for metal detection. Since DNA is naturally found as double-stranded, it lacks the necessary structural intricacy that we need to act as a catalyst. So they've been created in lab. The difference between what they are and, what a double-stranded DNA essentially is, and why I call it RNA-Cleaving DNAzymes, is because there's an RNA integrated into this strand over here. The difference is that the RNA has a hydroxide, and that hydroxide allows the stability of the DNAzyme to be a little less than what it would be if it was two DNA strands. So when the metal interacts with that RNA part, that RA right there, I'll get catalytic activity. Now, our DNAzymes have fluorophores and quenchers attached to 3' and 5', which you can see in this picture over here. Got fluorophore and a quencher. When my metal comes in and interacts with the cleavage site, the substrate cleaves off, and the fluorophore is no longer quenched. And that's my signal. The fluorophore cleavage that happens over rate of time. So this happens over time, as I mentioned. So the higher the concentration of metal, the faster the cleavage reaction is going to take place. So the more metal that I have in my system, the faster this reaction happens, the faster the fluorophore is no longer quenched. And that rate of reaction is what my signal is going to be. Get into the next slide. Now, here's some of the potential metal cofactors that can cause catalytic activity and are investigated in my work. So if you look into this in the literature, a lot of the times they just mention the free metals. So free lead or free magnesium, free sodium, whatever metal they're looking at. But with speciation and by using these in real water samples, which is ultimately what we want to do, we have other species that could possibly interact and cause catalytic activity. So here's a list of all the species that I'm going to study, that I have studied, in my project. And then I'll get into which ones that have actually caused catalytic activity and which ones have not. So the purpose here now is to calibrate the sensor. So the sensor GR5 in various systems where we measure the different parameters that can change the bioavailability. So essentially, the purpose here was to calibrate the sensor. And then remember when I mentioned that it's the weakly complex, so the free metal that is presumed to be bioavailable. So any metal that is strongly complex should not respond to my sensor because it's not bioavailable. We want this to happen in the DNAzyme, because if it doesn't happen, then you can't measure bioavailable metal anymore. Now there's factors that could alter the bioavailability. Some of the main ones are DOM, pH, ionic strength. They're the most important because they can mitigate the toxicity of metal. I want you to focus on one thing here. The metal bound to DOM, so anything that's bound to dissolved organic matter, is strongly complex and therefore should not respond to my sensor at all. So by studying my sensor and calibrating the sensor in all these different ways, using all these different conditions, we've got DOM, pH, we've got ionic strength. I'm hoping to calibrate the sensor and then be able to figure out what my sensor is responding to, what it's not responding to. Is it responding to bioavailability? Is it not responding to bioavailability? By looking into these factors, it's hypothesized that the DNAzyme metal signal will ultimately correlate to metal bioavailability. So they're just going to be directly related. Now, how did I do this? Here's my methodology. I'm going to try to go slow because it's interesting to me. It's kind of cool. And if you have any questions, obviously we'll get into that. You guys can ask questions later. So essentially you have the fluorophore, you have your quencher. I dissolve it in buffer A. You can ask me what buffer A is later on. It's annealed at 95 degrees. So I order the substrate and the enzyme strand from DNA Technologies. They give it to me individually. I have to anneal it myself. Once it's annealed, it's diluted down. And then as you can see, they're little strands and then they're double stranded. And then I go ahead and use a little plate. I separate them into plate. I add my DOC into it. I have controlled pH. I have controlled ionic strengths. And then what I want you to get from here is that when there's no metal involved at all. So in this case, there's no metal shown. You have a flat rate. So there's absolutely nothing going on, no activity, everything is flat. But this is what happens now when you were to add metal into the solution. So the left-hand side here, pardon me, this is what it looks like from my previous slide, flat, no reaction, to when metal happens. As you can see, you get an increase in fluorescent signal, so a rate of increase when you have metal in the environment. So here I have no lead at all. Then I have 50 nanomolar lead, so very little lead, 50 nanomolar. It's got an insane limit of detection. So 50 nanomolar, then all the way up to 1,000 nanomolar. So as I increase my concentration of lead in the system, the faster the rate of reaction is. So my maximum gets there much quicker, I think within 30 seconds, versus when I have less metal, then the maximum is reached much, much slower. I fit all this data to the first order rate law, and then I extract my K-obs, which is represented as K in this equation here. Now, what I want you to see is when I mentioned before is dissolved organic matter strongly complex as metal, therefore shouldn't respond. So that's what you see on the right-hand side here. So what I want you to take home from here is that as you increase the dissolved organic matter concentration, so the more organic matter that is in my system, it's going to bind to my lead, therefore less lead is available to interact with my DNAzyme, therefore, if there's less lead available, you should get a slower rate of reaction. So as I increase my DOC, I think below here is 8 milligrams per liter DOC, and this is 0, super fast, super slow. And you see that pattern, and you see that pattern as you go along. So KOBS, which is my analytical signal here, is directly related to lead species concentration. Then, as I mentioned, I want to know, well, what is it actually responding to? Is it just lead? Is it other species? What are the species? So I I fit all my data, all those, the table that I showed you, all the different parameters that I had mentioned, fit all the data, determine my KOBS. I used a WAM predicted model where I can predict to see what is my species, what are my output species, when I put the exact same parameters into the computer system. So calibration here is a function of all the organic species that respond to my sensor. So as you can see, this is the calibration equation right below here. And there was no individual species that was responsible for activity on its own. So it just wasn't the free metal. It was actually a sum of the 3 most important species, which are shown here. So my sensor actually responded to the lead, free lead, to lead chloride, and to lead hydroxide. All these other species, lead acetate, lead carbonate, lead bicarbonate, lead bound to DOC, and the total, the total lead should also be circled. None of these species are responsive to my sensor, which was good in my opinion. So I took that data, I took my responsive species, and then I compared it to predicted species. So you have your measured on the x-axis, and you have your measured on your y-axis, and your RAM predicted on my x-axis. So what I predicted versus what I measured, my one-to-one line, and you can see that all my data agrees fairly well. Actually, it agrees perfectly, meaning that my sensor was responding to bioavailable lead, and it wasn't responding to stuff that was not bioavailable. Now, what does that do? That means that I no longer have to do all these other measurements. I don't have to measure temperature, pH. I was able to solve the bioavailable metal with just one measurement, and I just had to solve for what I wanted to. In that case, I solved for the free lead in the previous equation. If I want to solve for other species, I can do that as well. So future work here, what the bigger picture is. Currently, modeling is done through Bioavailable Model for risk assessment. In order to use a Bioavailable Model, we have to input all these measurements like I mentioned. They cannot be done at the site. After measurement, bioavailability is then only predicted. In addition, instrumentation exists that can only measure weakly complexed or free metal, but we know that it is only the bioavailable metal that is toxic and thus the most important. So instead of measuring these 7 different parameters, waiting days for results, we replace it with one measurement using DNAzymes. Now, risk assessment managers and regulators need this measurement in order to regulate the levels of lead entering into environments. They already require the bioavailable lead measurement to make decisions. However, instead of doing it the old-fashioned way, they can do it this on-site in real time and make decisions much faster and much more accurately with my one-step measurement. So I'd just like to take the time to thank everybody, my funded by Global Water Futures, NSERC, all of you guys at SCWIST, all the judges, the audience, the panel, my professors. Thank you very much for this opportunity. And now I look forward to taking questions from all of you.
Okay, thanks, Gagan. A really good presentation. So I will leave it now to judges, Brittany and Anjana, for panel discussion.
I'll ask the first question maybe. So you mentioned at the beginning that you're looking at specifically lead, but there are other metals you could also look at. Are there any others that you're interested in for your specific project?
Yes. So, I mean, am I interested in other metals? Not really. But we were going to look at silver prior, except when I thought that using the DNAzyme would be a lot easier and I'd be able to get ahead. It wouldn't be so many halts that we came across. If that never happened, then yes, we'd be looking at other DNAzymes like silver. The reason I mentioned silver is because the DNAzymes are fairly new. I think the first one was 1994, and it was the GR5, which is why I worked with that one. But there's one that's AG10 that's fairly new, fairly older than the other ones. So I was like, well, if AG10 exists, it's been studied already, we can just buy them and use them and apply them, then why not use silver as well? Except when we put these in real water samples and then trying to figure out how are we can actually get our analytical signal, what our analytical signal, what we wanted it to be, sort of working with that, we kind of went much more deeper with lead than didn't even bother looking at other metals. But they do exist. Actually, a lot of these exist, and they're very, very specific for metal. So the GR5 doesn't respond to any other metal out there. It just responds to lead. But you can find ones that respond to multiple metals at once. So yeah, for now I went with the GR5. It's become my baby. But maybe in the future, I'd be interested in seeing how the other ones respond and if we would take it the same way as we did with this one.
If you don't mind, I'll ask one follow-up just because she touched on something I was going to ask next. So did you challenge your GR5 with any other metal just to make sure there was that specificity, or did you base it off of, you know, the research that's been done and that it's been proven?
Me specifically, no, but Juan Liu, my professor at Waterloo, he is the one that actually studies these in vitro and then alters them conditions-wise, depending on how we want them to respond. You can actually do a lot of things to these things. You can change functional groups and make them respond in a different way. You can, you know, mine actually originally was a much longer strand, but then we shortened it in order to increase our rate of reaction. I didn't need it to be that long. 30 seconds was enough for me. Mm-hmm. But I could make it longer. I could change the functional groups from PO to PS. I think that's I did go and learn from him. The students did teach me. So yes, we did study them in different metals, just put a bunch of different metals in there. Didn't respond to anything except for lead. Now, when I'm doing these in real water samples, when I mimic the conditions, I do have other metals in the system. They didn't respond to anything except for my lead. So yeah, and like I said, this one's been significantly studied, and they've been studying different metals as well. It only responded to lead, which is great.
Okay, perfect. Thank you.
Hi, Gagan. You did a wonderful presentation. Thanks for that. I think this is a little bit fall on, or you test base this. So my question is, have you tested real water samples close to mining areas? Have you done those testing? So along with control?
So currently, no. I mean, before COVID happened, I did take water from the sink that's like in Waterloo, and I just threw them in my sensor to see what would happen. I did get a response. I just want to make sure nothing crazy was going on with my water. But that is the future work. So currently I'm doing toxicity tests at this point in time to confirm that in addition to my WAM predicted, so modeling, does my DNAzyme actually respond to bioavailability? And the best way to do this is run a toxicity test. So I'm doing that at the moment as we speak to make sure that this is what it's actually responding to. The next step actually come January, based on my project outline, I am going to be using real water samples. We decided to go with lakes near our area. But maybe in the future, yeah, definitely getting water from sites that have a lot of lead in them, such as mining sites, would be an excellent way to go.
Yeah, I think you should also consider looking at agriculture field close to, rivers close to agriculture fields, because there are a lot of pesticides which they have used earlier or using. So that is also will be a very good place of collecting samples and testing. For sure, yes. And will be a good data.
Yeah, absolutely. Yes, thank you.
Thank you very much. Yeah, I'm good, Brittany. Yeah.
Okay, I'll ask another question then. So you mentioned this WAM predicted model. Can you just maybe walk us through what exactly that model is for people who don't know?
Yeah. So the WAM predicted model essentially is like the biotic ligand model. It's a model where you can input all your different parameters. You can input, like I input my pH. I can put my salt concentrations. I can just mimic my exact experimental setup. The cool thing about WAM is that you can actually implement dissolved organic matter as well. Biotic ligand model, there's still an output that has not been incorporated into all the biotic ligand models. So WAM, I can input my dissolved organic matter as well. So essentially what I do is I input all my parameters, and then it spits out the values for me as individual species. So I use that as a confirmation as to what's actually going on in my system. So that's what it is. I use Cheeks, which is a free version of WAM, because I don't really need the complicated version of WAM. I'm not really doing a lot of crazy things. I just implement I actually do it first. I'll implement all the conditions into that system, gives me a bunch of values, tells me this is going to work, this is not going to work. Because I have figured out the limit of detection of the sensor too. So if it's going to give me crazy values, I know not to do it. So it saves me some time. So that's the model I've been using as I go along. I've used it very, very often. It's a good model. You can just download it online. So that's essentially, that's all it is. It allows you to input all those parameters, and it has organic matter implemented into it, which was the key thing for me, because it is a mitigating factor for bioavailability.
Perfect. Thank you so much.
You're welcome.
When are you expecting this project to be completed, and have you published papers on this?
So I'm actually doing a proof of concept paper right now, because that's where my sensor has led to. That was the first thing, is proof of concept. So I think we've been fairly confident in proving the concept that yes, this DNAzyme is something that can lead to a sensor for bioavailability. So once that paper is published, I'm actually in the edits phase right now. The next thing will be a toxicity thing, which is happening, and then it will be real water samples. Once we've done all that, then it'd be something that we would, take further, look at a way to implement this into an actual portable sensor. And then that portable sensor will be something that we would sell. Now, I'm in my last year of PhD, so I don't know how far I will specifically get. Maybe I'll stick around for a postdoc or not with this project, or maybe the next student will take over. I don't know. Maybe I'll find another sensor to work on. I don't know, since I keep finding new things. So we'll see.
Yeah, this is a very, very interesting sensor, and I hope you will get in touch with some industry and get this product to the market, or get this product to the place where it needs to be used, like testing areas and all. So
yeah, for sure. I mean, I always see people, like, for us living in Waterloo, we actually have to take our water, because there's a lot of well water here, to public health, and they have to measure lead levels, and they have to measure, they have to determine if the water is safe and what's in it. So imagine just being able to do it at home. You don't have to take your water in and wait a couple of days to get results back. So it's a wide range of application and where it can be used, for sure. Yeah.
Yeah. I hope your work will be used in future.
Thank you. Me too. Me too.
Because Canada is looking for these kind of projects coming from lab to the industry to the Canadians. I hope this will reach Canadians at one point of time. Yeah. Thank you.
Thank you.
Okay, any other question, Brittany? Do you have any other question?
I'm okay.
And Anjana?
I'm okay, Noeen, thank you.
Okay, so I will start taking questions from audience. They have actually lots of questions.
A lot of them, yeah.
But I will just ask the questions. I will leave it on our judges to evaluate the answers. Our first question is about any prospectus using these sensors in wearable devices. Do you know any prospectus of using these sensors where we can use these in devices, wearable devices, I would say? That's the question in the chat box.
I'm trying to read the question.
Let me actually send you directly, Gagan.
Yeah, please. So I don't know. I visualize better than hearing it.
Yeah, so you can, let me see. It is about wondering any prospectus using these sensors in wearable devices. I think it's more about biocompatibility.
Okay. So the DNAzyme itself is a sensor, in my opinion, on its own, depending on what you're using it for. Now, I don't make these DNAzymes. I took the DNAzyme concept and I applied it to my situation. There's tons and tons and tons. I think there's millions of ways where you can use this DNAzyme because of how easily you can manipulate. You can change the DNAzyme the way you want it to. It's extremely stable. It could be used in many different environments, and they can be used many different ways, because biosensors on their own, there's calorimetric, there's electrochemical, there's fluorescence, there's many different types of biosensors. Depends on what you want to use this for now. Can the DNAzyme be altered into different devices? Yeah, absolutely. Depends on what kind of device you want, what you want to use it for. Now, I have seen people use DNAzymes for measuring things in blood serum. They're measuring mercury levels in blood. So they're using this DNAzyme in that as well. I've seen the DNAzymes be used for measuring, like I said, for my situation, different metals in different environments. The DNAzyme itself as well, like depending on where you want to use it, people have done all crazy things. They've coated it with gold to stabilize it. They've put it in different capsings so that you can take it into different conditions for stability reasons. So yeah, there's lots and lots of potential of using these sensors in different devices. Have they become portable devices yet? No. I think people are still using this as a concept, proof of concept. Sort of that's the stage that they're at. Do I see these going into, wearable devices? Absolutely. How long it'll take to get there depends on what someone is focused on. Do I think that this could be used for something like sensing COVID? Yeah, maybe. It's a DNA. So depending on what you want to use it for, using genetic coding, implementing it the way you want to implement it. And then again, just using different ways to stabilize the DNA on its own. You can use this for all different things.
Okay, thanks, Gagan. I will move to the next question. The next question is, how would you verify that this is responding to a bioavailable fraction of metal?
So that was the thing, is we verified it one way, which was using the WHAM. I had implemented all these parameters. WHAM spit out a number for me. I compared my predicted versus my measured, and it was a very good comparison. The next way would be to toxicity tests. So we will do a toxicity test, and the toxicity test essentially is a bioavailable metal test. Whatever is available for the species to take up, it will take up, and then we'll find our EC50s or EC20, depending on whatever we're measuring. In my case, I'll be doing EC50s. If those numbers are identical to what I measure or close enough to what I measure in my DNAzyme, then that's how I'm going to verify that it is measuring bioavailable metal and not something else.
Okay, thanks, Gagan. Judges, do you have any follow-up questions from these questions, or you are good? Okay, so the next question is, can you please share a bit more about the practical applications of this test? I think you already talked about using it in different sensors. If you can just name 1, 2, or 3 most important. I don't think you have to go into detail of this question.
Yeah. So I'm going to talk about things that I like in terms of where I would like to see this stuff happening. And usually to me, it's fixing real world problems. And the one that we're facing right now is a global pandemic. We had this huge thing where we're held behind because we have to find a way to detect the virus. I built a sensor in my master's, ended up patenting and stuff like that. It was electrochemical sensor. We used carbon nanotubes, and we were able to sense bacteria. The DNAzyme itself is a DNA sequence. That's what it is. It's a sequence that's specific for lead. You can alter the sequence to what you want to alter it to. You can change the sequence to something like COVID. And then, because it's so specific for what it's altered to, you can see this being applied in situations like that, where you would be able to sense something like bacteria, a virus. In the case that, most recently I went to a talk was mercury in blood, because we have mercury toxicity. So again, it just depends on what you want to work with. I'm trying to emphasize here is that DNAzyme is so unique. It's such cool technology. It's got tons and tons of potential. You just have to study the DNAzyme, and there's tons of groups out there that are studying it. How do we alter these to the way that we want to alter them? And what are we altering? Do we need to change the whole sequence? Are we changing the loops? Because it's that catalytic loop that was specific to metal. What do we need to do in order to have these in application? So mine right now, COVID, using it in water samples. Again, I see people go to public health all the time. We got to take our samples to public health and get measurements back. I just bought a water softener, and I didn't even know I had to know the lead level if I had a lead problem. Had I had known that, I would know what to adjust my water softener to. There's so many different things. And then third world countries, they have so many issues of having access to clean water. Imagine just being able to have these sensors out there, and they could just, be able to test that water immediately, get a response right away, and then be able to drink that water, right? Hikers, a lot of hikers would like to use this. I remember going to a conference, and there was people that like to hike, and they don't know if the water that's coming down the river, they can drink it or not. Having something like this, and like I said, you can have sensors where implement many DNAzymes. You can have DNAzyme that senses lead. You can have a DNAzyme in there that senses a different metal. So instead of just having lead respond back, you have all these other metals respond back. You got this, and this, all in this portable sensor. And then what levels, are there and what you can drink, what you cannot drink. So again, a lot of potential. Again, this is just early stages. Maybe in the future, I'll be back and I'll have something else for you guys.
Yeah, sure. Gagan, we would love to hear more about it in future, how far it went. Okay, so the next question is about DNAzymes, if you can tell, like, why are they called DNAzymes for general public? Like, I am not biotechnology person. I have little bit molecular biology background, but for me and for other people who have IT or other backgrounds, so how can you define why are they called DNAzymes in an easy wording for them so that everyone can understand?
Yeah. So a DNA or DNA is double-stranded DNA. Usually you have substrate strands. The reason why they're called DNAzymes is the NAzymes, the N and then azymes, is because the enzyme strand is what's modified with an RNA. Because that enzyme strand is modified with RNA, they came with DNAzymes, just emphasizing that it's the enzyme strand that is modified, hence DNAzymes. Everything is DNA in there except for that enzyme strand, where they pluck out that one strand and they implement an RNA in there, and then hence you got your name DNAzymes. That's where the name comes from, just altering that enzyme strand, that's all.
Okay,
Yeah, I didn't come up with that name. It's just a name that someone came up with back in 1994.
The next question is, what's the difference between DOC and DOM?
So DOC is dissolved organic carbon. DOM is dissolved organic matter. It's actually dissolved organic matter, but DOC is the measurement. So it's dissolved organic carbon milligrams per liter is measured as dissolved organic matter. So it's just the units, I guess, is what you want to call it for dissolved organic matter. That's what it is. I just like DOC because it's all in my head as measurements. So, but it's the same thing. It's just a measurement of DOM.
Okay. Okay, thanks. Anjana and Brittany, you have any follow-up question from these questions though? No? Okay.
I can ask one.
Okay. So Brittany has one question.
I know you mentioned that, you haven't tested these real water samples yet, like in the field, but is there a major challenge that you can foresee using your technology in the field, or do you think things will go well? I mean, you don't want to think about the negative, but is there a challenge that you foresee on the horizon?
So my test samples now mimic real water samples. Obviously, it's a controlled environment. I don't have all the other crazy things that'll probably end up in water, when I go collect water sample. But in terms of the pH, ionic strengths, and all the other things, all the salts and all that stuff that end up in water, my samples right now mimic real water samples. The challenge, and I've got this question a couple of times before from the biologist's point of view, is, well, what about other like amoebas, other bacteria eating things? Is a DNAzyme going to be stable? The DNAzyme is extremely stable. I don't see a stability issue coming into play. And if the DNAzyme does only respond to lead, it shouldn't respond to anything else. The only challenge I could see happening is those unknown factors that would sort of interact with my lead, and then me trying to figure out, well, where is my lead going? How much lead is actually in there and how much is actually interacting with my DNAzyme. And if it's something different than my controlled environments, well, where is that lead going to? Other than that, and other than maybe my DNAzyme coming in contact with bacteria or something that's going crazy and eating it up or something like that, which again is a crazy idea, but it could happen. I don't really see a lot of other major issues coming into play. And then of course it's figuring out a way of how am I going to, get my analytical signal and what it's going to be. And is my equation going to be able to be the same? What else am I going to have to implement into it? It's figuring out those things. Other than that, I don't really see anything happening, but again, who knows? We'll see when I measure. Maybe it won't work. I don't know. I hope not, but so far it's promising. Which is good.
Okay, thank you.
Yeah.
Anjana, do you have any follow-up question?
Yeah, no, sorry. Yeah, whatever coming up, so it's discussed very well. Like Brittany is bringing up, and in the chat, audience are very actively asking questions. So I'm good. Yeah, thank you.
Okay, I have question from audience. Will the DNAzymes be stable in natural waters?
Yeah, that's
What
a good
is the
question.
range of chemistry? Do they work? And freshwater, saltwater, like how would you differentiate both, like by taking these two examples of salt and freshwater?
Yeah, good question, Scott. So I'm glad you asked this because this is something if I had time, I would emphasize in my presentation, but I didn't have a lot of time. I did actually study the range of the sensor. So that was one of the main things as proof of concept is where's the sensor going to work? Obviously I don't think the sensor is going to work because of stability issues. I don't think it's going to work in anything I put it in. So what I did was I mimicked, again, like I said, saltwater, freshwater samples, where's my sensor going to work? Currently, it works in freshwater samples and it will work in saltwater samples. Now, my DNAzyme actually requires a certain level of salt in order to be stable. And what I mean by stable, I mean that cleavage site is blocked at the moment with salt. But if I decrease that salt concentration, my cleavage site is open to anything. Now, because my signal is rate, I can only measure with the human eye, and by pressing start on my instrument, I can only measure so fast. If my DNAzyme is too fast for me, which I've had come in issue with, where my ionic strength is too low. So I think 10 millimolar was the lowest I could go, not because the DNAzyme was unstable. It was because for the human eye, for rate of reaction, I wasn't able to actually sense anything below 10 millimolar. It was just too fast. I wasn't able to get those initial rates of reaction, which is my analytical signal. If there's a different way to get your analytical signal or a different analytical signal people want, of course, you can adjust the parameters that way. But for me, I ended up with a barrier there that, okay, I need this much salt in order for me to actually even get a reasonable rate of reaction where I can actually take it forward and be able to measure something. My boundary, though, is on the other end as well, is, well, what if I had too much salt? So this sensor actually works better in saltwater conditions because I have enough salt that can control that catalytic site. It can control the rate of reaction where I can actually sense stuff, where I can actually make sense of what I'm actually picking up. Same thing with pH, again, because it adjusts speciation, lead hydroxide was my most responsive species, surprisingly. And at basic conditions, I'm going to have more lead hydroxide. At acidic conditions, I'm going to have less hydroxide. So in that case, that in basic conditions, my rate was much faster. And in more acidic conditions, my rate was slower. So it's, well, how fast can you go and how slow can you go? So in terms of answering the question in a one sentence thing is, yes, the sensor has barriers. Yes, it's got limits. It does work in freshwater. It does work in saltwater. I'm lucky enough to be able to control different parameters at the moment, different pHs, different salt concentrations. The sensor will have to be altered in some way in the future, depending on what environments you want to work in. But right now, for freshwater, saltwater conditions, the sensor is doing fairly well because I'm able to have those things that I need in order to control that rate of reaction right now.
Thanks, Gagan. Brittany, Anjana, any follow-up question?
I have a question. So you mentioned, right, that the current methods, they're expensive, complicated, time-consuming, and then this way we're, decreasing expense, less complicated, and then you also mentioned third world countries. So from an expense point of view, is it something that's going to be accessible to a third world country, or would it be an expensive piece of biotechnology to take there?
Good question. So from my point of view right now, with the most knowledge I have based on where I am in the project, the DNA costs probably $100, and I think I've only bought $200 worth of DNA because I've messed some stuff up over my last 4 years, and I've been able to run so much experiment because I only need 1 microliter, like a very, very small volume of DNA. So yeah, it's feasible in that sense. That's all I've done so far. Now, to make it portable is something in the future, obviously keeping in mind as well, if I want it feasible, how to make this into a portable sensor that it's still feasible. The concept on its own is fairly cheap. DNA is cheap. The salts, it's salt, it's cheap. Real water samples is free. And then I use a fluorescent meter, a fluorescent spectrometer to measure my fluorescence. So, but there's ways that we can use it into a small portable sensor. You just get fluorescent signals. You can do electrochemically, which is even more cheaper. You just get an electrochemical signal when it detects something. So in terms of expense-wise, I do think that this is a feasible way for sure.
Perfect. Thank you.
Anjana, you have any question, follow? Or it's good?
No, now it's good. Yeah.
I think that this question actually covered other questions too, like final applications and commercialization. that is always the question for research projects, how widely it can be implemented, So I think I will take just one last question from audience. Maybe it will be good for information for everyone. How would you reset the sensor DNAzymes in a practical device?
So that's a good question. Obviously, because it's like, well, is the sensor DNAzyme reaction a one-time use? Do I have to, reuse DNA again for the second time? I'm actually trying to work on standard additions method right now, where I would use the same DNAzyme, put my sample in, take a measurement, put more metal in, take a measurement, put more metal in, take a measurement, using the same DNAzyme. Does it eventually, max out where the DNAzyme no longer has any space for binding to happen? Maybe. I don't know that limit at this point. It's a study that I'm still conducting to see as well, what has it reached its saturation. I think so. I think it would have to, based on practical and the chemistry. Is that, well, if it's binding, eventually it's going to stop. It's going to bind and then be done, right? How would I reset? I guess you would just need more DNA. That's how you reset. I don't really see a reverse reaction happening, but maybe there's a way to do a reverse reaction and then get your measurement, have a reverse reaction, and do it again. So I'm not quite sure how that would happen in the future. I know there's a lot of future questions which were brought to my attention today. I'll keep thinking about them, and maybe there's a way to sort of maybe use some kind of like pump that just like sample and then it goes and sample and it goes and then eventually saturates and then you have a new DNA. I don't know. We'll see.
Okay, thanks, Gagan. So there is only one question. I think I can answer that, right? Only what your research seems very promising. Just eager to know what stage are you at your PhD. I think you were in last year of your PhD, right?
Yes, I'm in my 5th year, so I'm actually doing an extra year. I guess COVID slowed things down, and we all got lazy and all that. But this is my last year. Hopefully, things go as planned, and I can get something out there useful. But yes, this is my last year. I don't think I want to go any further than that.
Okay. Cool, thank you. So I think we covered almost all questions about sensors durability, what are the DNAzymes, DOM, applications of your project, which of course you already talked so many times, how to use in different devices, practical applications, and proof of concept. I'm not sure if we can take it. So would this be referred to as a proof of concept? I think I will take this question.
Yeah,
and
so this.
Yeah, so the rest are all done. So I think, this is, I think, the last question for you.
Okay. So yeah, this first publication that I'm working on right now is the proof of concept paper. It's proving that this DNAzyme has not been used the way it's been used ever, the way I'm using it. And it does show promise. So it is a proof of concept paper. I got a bunch of information on the sensor limits, what the sensor has done so far, what it's proven, and it'll probably have a section in there that's like, well, what else could it do? It tells you the species that it responds to, which gives information to those biologists that are not sure what bioavailable metals even are. We had that hydroxide, we had different species. And it also tells those DNAzyme workers that because if you read these papers in literature, they just talk about the free metal. They mentioned that it's just lead 2+ or it's just magnesium 2+ responding, but it's not. In our case, we've shown other species that also respond to this. So it is a proof of concept paper. This is what it is. And then hopefully the next paper in Toxicity will be an even bigger proof of concept. Look, it is what it is, what I've said before. So that's what it's at. And in terms of, I know everybody loves applications. I told myself I was not going to do a project that did not have a good application. So it's just not, I don't have that sort of interest. It doesn't drive me. I need something that's going to solve a problem that we have in a way that I can visually see it going somewhere in the future. So that's why this is it's a hefty application project for sure. Yeah.
Thanks, Gagan. I think that's a very good answer. It's same as with me. Most of the time when I work as a researcher, I would like to focus more on the projects which have broader scope and more applications, and I can see some growth or a trend in the next 5 years, right? So you would like to work on the tried and repeated trial errors or something. I don't know if I'm saying
it's just, I don't think I'm smart enough to do something that doesn't have an application. My brain just doesn't go there. I'm like, what am I even looking at? What is this? Like I mean, if anybody's doing something that's, kudos to you guys. We obviously need those scientists out there too, but it just wasn't for me. It had to be application related.
Okay, thanks, Gagan. And Brittany and Anjana, I would leave it to you to say some closing sentences, and then I will close the session at the end.
Sure. I just want to thank you so much for your presentation. Actually, it was something I didn't know very much about. And when I saw your presentation, I actually read a bunch of papers this morning.
Oh, nice.
So
Okay.
I'm like that. I still search PubMed on a frequent basis. So it was really nice, and you explained it very well. It was easy to follow, and I really enjoyed, and hearing your passion for your field is just amazing. So I just want to thank you so much for your presentation today.
Thank you very much. I appreciate that.
Yeah, I second Brittany. So same thing. So very interesting topic. And I also did a lot of research, learned many things about this topic. And I'm really looking forward to get this product to the market. You shouldn't leave it at this point, because the Canada always promotes, and lots of funding agencies are there to help you. So you should get this product to the market.
If you want to hire me after, I'll bring it with me and we can just work on it.
Yeah, absolutely. Let's work on it.
That's wonderful. Okay, yeah, I think I will be actually talking about student internships too. I would like to talk with different sponsors and like our sponsors of Symposium for sure. And then I will ask them that if there can be internships opportunities, especially for the students who were the finalists in our Symposium. So I hope it can work out, and then you can get somewhere and you can do the project which you want to do,
Yeah.
Yeah, I would like to say thanks to all of you. I hope you enjoyed this session, and thanks for being here. And I know I was continuously moving. I cannot sit calmly at all. This is not my nature. I need to move to survive. And I will share now some closing slides, and then I think we are all good to go. Over here, okay. So if all of you can see, then I would like all of you to follow us on Eventbrite. Our all sessions are already live there. In addition to these sessions, you can always find other events which are coming in SCWIST, which are like workshops. For example, we recently had about video editing workshop, and we are getting even some AMA series with some sponsors, where sponsors will bring some recruiters from their company, and they can ask questions to the students and audience. And okay, can I go? Okay, yeah, no. Okay, the next event is going to be, the next session is going to be on June 16. It's Wednesday. Tofa would be presenting with us, and she is from ANRS Institute. It's Institute Armand Frappier Health Biotechnology of Canada. And if you would like to register, go to our events page or go to the Eventbrite and register it for free. And we hope to see you soon in the next session. Also, one more thing, that we update our website almost every week, and we are also going to update our catalog every month. So for our finalists, for our sponsors, for the judges and committee members, whenever you need information, you can also keep visiting our website for more information, or write me an email. With this, I think I will end this session. I would like to say thanks to all of you once again, and thanks to our judges for being here. I mean, it could not be possible without you, for sure. And also it could not be possible without Gagan, like students like you who could not participate. Then session could not happen, right? Have a wonderful day. Take care. Have a wonderful evening. I will upload this video, recording of this video tonight on SCWIST channel, and I will share this link with our finalists and our judges and other committee members. Thanks, everyone. Write me an email if you have any questions, and I will let you go now. So take care. Have a great day. Or evening.
Thank you. A big applause to Gagan. You did very well with all your your effort was wonderful. Thank you. All the best.
Thank you.
Thank you, everyone.